pointer is this one. All right. Well, thanks everyone for coming back, and we're going to start something a little different now. I'm very pleased to be able to be the kickoff speaker for this session on endemicity of FMD. And as I started to think about what kind of talk to put together, I was struggling a little bit and thinking about work we do on persistence in the laboratory and work we do on FMD persistence through field studies with international collaborators. I couldn't quite choose which one to focus on, and so I sort of thought about this trend, this evolution of these disciplines of study that we have inherited and how they relate to one another, how the field studies and the laboratory studies play off one another, benefit from one another, and bring us to this greater understanding of the carrier state of FMD. And so I decided to sort of combine them into a single talk, and without really presenting any new data, without giving you any strong conclusions, I'm just going to talk about the tradition that we have inherited, a little bit of ancient history and a little bit of more recent history to talk about our current state of knowledge of the FMD carrier state and sort of break it into some nice compartments of areas of understanding or lack thereof. So the 10 years between 1959 and 68 were a period that a lot happened in the realm of FMD persistence and defining the carrier state. And so these are the landmark papers, and if you haven't seen them or read them, I encourage you to go back and look at them. They really provide a great basis for what we're doing today. And it's amazing when you look at these papers that these guys were asking questions then that in a lot of ways we're still trying to answer today. So I'm going to step through these papers a little bit and use them to bring us into the current era and try and tie it all together. And so the Van Beckum 1959 paper is the one that is most often referred to as the paper that defined the carrier state. It is an experimental study. It is the first time that someone looked at the prolonged shedding or detection of FMDV in infected ruminants over a period of 5 months or greater, and it set the stage for future studies to come. What's interesting is you read that paper and you see the extent to which this guy back then was already struggling with things that were happening in the field and in the laboratory, and he was already sort of in the same mindset that I'm describing to you right now, that we need to integrate these distinct disciplines and use them both to move forward with our understanding and categorization of the carrier state. So I think a lot of us are laboratory-oriented scientists, and we're pretty comfortable with the types of advantages that we have with those types of studies. Just going to talk about it for a minute. Prospective study design is clearly one of our favorite things. The fact that we can pick a virus, we can pick an exposure condition, challenge route, time of challenge, dose of challenge, all of those things, and that we can deliver this virus to animals and we can monitor them in this prospective defined manner with very intensive sampling, and that we can minimize to the greatest extent possible all the confounding factors that can challenge interpretation of data in an experiment. Typically, when you're doing a laboratory experiment, you generate your samples in some sort of animal facility, and within an hour at longest, you're back in the lab and you're freezing down these samples. You have tremendous control of sample handling and cold chain and preservation of the integrity of those samples. You have reduced operator variability because you define who's going to be doing the tasks. You have the same guys or gals doing those tasks day after day. They become experts, and you know who you can count on for particular aspects. And in our hands, it's pathogenesis studies in our laboratory that we're doing most often and most intensively, but similarly vaccine studies and other aspects of study of FMD are also similarly suited to this type of experimentation. Of course, there's limitations as well, and we'll get to those in a little bit. But so let's think about this concept of lab experiments with the subheading of detecting and defining carriers. So this is a little bit of data. This is published from 2016 in a JVI paper, and this is just antemortem dynamics of animals, cattle that were exposed to FMDV by the intranasopharyngeal route. And we're watching them here. We're watching the dynamics of infection over time on the x-axis and detection of FMDV on the y-axis. and we're seeing nasal swabs and saliva in green and blue, and then starting around 14 days, we start doing probank sampling. And so again, just this intensivity of sampling, the granularity of the data you get, you're just never going to get that when you're doing field studies, and it provides some tremendous advantages. And so in this model, we have been able to define and identify carriers at 21 days. This is in unvaccinated, naive animals. And when I say we identify them as carriers, my point is to point out that from the point of 21 days forward, animals are probank positive, they stay probank positive until they get out to the conventional definition of 28 days of the carrier state. The other animals that are negative, probank negative at 21 days, they stay negative. That therein is our basis for saying we can identify carriers at 21 days. This little note is a reminder to me to point out that in these types of experiments in our laboratory, if we're detecting probank positive animals, we have good concurrence between PCR and VI. And so there's, I think, a lot of reasons for that, but it is uncommon and uncharacteristic in my experience when you go to field studies, probanks that are generated in the field, you don't typically have that finding. At least I believe that is the case, that most people find there are a whole lot more PCR positive detection of FMD vRNA than there are isolation of infectious virus. And it is, again, another one of the advantages we have, which I think is very closely related to sample handling and cold chain as we do in the lab, as compared to all of the potential failures that occur from the time you collect the sample in the field till you have it in the laboratory, as Don King mentioned today in regard to outbreak. It's the same but worse with probank samples, which have lower quantities of virus. So Suttmoller, 1968, this is the paper that defined 28 days as the cutoff for carriers. And as you read this paper, you realize that it really was a completely arbitrary definition. It had to do with how long he could keep the animals in the experiment. And it turns out, very conveniently, it is pretty close to what we find experimentally, but it was a definition that came from convenience of 28 days. So here's a look at vaccinated animals. I mentioned that unvaccinated animals, we can differentiate carriers from non-carriers at 21 days. In vaccinated animals, we can see it even earlier. So as early as 10 days after challenge, here we have the animals that have diverged towards the carrier state, and here are the animals that have diverged towards clearing or being terminators, as we like to call them. This is a reminder from Dr. Murray et al. That not all creatures are created equally, and so the Cape buffalo has been shown that oropharyngeal fluids collected by the probank are not the most sensitive sample to use, that actually these tonsil swabs that they do under sedation, right, as I understand it, are actually more sensitive technique. And moving forward, when we start to think about small ruminants and other wild species, we also should be aware that one size may not fit all for that aspect. So limitations of experimental studies. There's the practical burden, just the cost of maintaining animals in a containment laboratory becoming more and more expensive over time. Animal welfare concerns about doing experimentation on animals is also a concern. And no matter how fastidious we are in designing what we think is the perfect experiment, you get up and deliver it, and someone's going to say, well, why'd you pick that virus? Why'd you challenge by that route? Why'd you feed those animals alfalfa cubes? So every choice that you make in these experiments, it seems to be the right choice in some ways, but it is still artificial and is not a representation of what's really happening in the natural environment, no matter how closely you try to replicate natural conditions. So this is another recent paper that took another approach to defining the carrier state, and this is from Bronsvert et al. And the point here was that taking a probang and throwing it onto some BHK cells is not necessarily the right way to define whether an animal is a carrier or not, but rather there's a probabilistic approach based on statistical models. And it definitely is attractive in many ways, and I think it provides a very novel approach based on field data, in this case, to define carriers in a different manner. And to see that over time, as we've come to expect from other studies, you can predict, in this case, a decrease in the amount of carriers that you would find over time, with time being months since the last known infection or known outbreak. So field studies. That study I just mentioned was based on data collected in Cameroon in a natural endemic setting. And so advantages of field studies, they are undeniably linked to reality. They are based on naturally occurring disease. The scale and the scope of these studies is essentially unlimited because FMD is going to be out there for as long as we're able to go out there and study it. Just a matter of how much resource you can throw at it. Enables associating naturally occurring events with naturally occurring background data to the extent that you can define that data. And you can make the spatiotemporal relationships with the same degree of, correlated granularity based on the amount of data and type of data that you're able to collect. But there are limitations to field studies as well, of course. And so it's an unknown context of infection. You have not controlled the environment. You have not controlled the exposure of the animals to the virus. And you don't know all of the environmental factors that have contributed to that slice in time that you're out there collecting a sample. And often that information is limited. The outcomes and conclusions that you reach are still limited to that particular context. And so you have a project that is based on naturally occurring endemic disease in Vietnam. To what extent are you going to be able to translate that to natural occurring endemic disease in Botswana or India or Pakistan or anywhere else? And that, I think, remains to be determined. Technical challenges associated with field studies. Again, cold chain, I think, is hugely important in the conclusions that we're able to reach, the quality of samples that we're able to bring into the lab, and the complications of transport, getting those samples from the field back to your laboratory. Limited capability, there's no way you can do daily sampling and the type of intensivity of sampling that I was showing for laboratory studies. And so thinking about these two different ways of defining the carrier state, I think they each have their advantages and their disadvantages. I wouldn't want to go out into the field and make decisions about depopulating a herd of cattle based on either one of these. But I think moving forward, and this is sort of what I want to get at with this presentation, is that as a research community over time, we're going to have to learn how to unify and combine these different types of approaches to come up with ways that are sensible to deal with these carrier animals that we have not yet defined. So sites of infection and carriers, another area of active research that's come mostly from the laboratory. Burroughs in 1966 was the first guy to identify the nasopharynx as the site of persistent infection. And that's not time for me, is it? I'm okay? Okay. Halfway? Okay, good. Thank you very much. So Burroughs told us that the dorsal soft palate and the roof of the pharynx are the sites of persistent infection. And just to pause for a moment and clarify something that is not always clear amongst people that I talk to, this is the nasopharynx. It's not the oropharynx. And so it's an inconvenient truth for us that we collect oropharyngeal fluid when we pass a probang through this oropharynx into the esophagus. But the sites of infection have been shown over and over again to be in the nasopharynx, not the oropharynx. So in our laboratory in recent years, we've taken this a little further than Burroughs. And so we've used different techniques to localize the virus. And here, these red cells are tagged with anti-FMD VP1 antibody, and we're showing the localization of the virus on the surface of the nasopharynx. And that, in slight contrast to the work by Juleff and Dr. Charleston, that in 2008 they put forth this description of localization of FMDV to the follicular dendritic cells and lymph nodes in the carrier state. And here's a very nice co-localization in submandibular lymph node at 38 days after challenge. And this has gone on to be something that we have seen again in the recent paper from Murray et al. And other incarnations from Pirbright. And it's a very distinct yet novel and interrelated approach to what we've shown to be the sites in the epithelium of the pharynx. And my point is to point out that these are not mutually exclusive findings. I believe that the reality of the carrier state is such that there is virus moving back and forth between the epithelium and the lymphoid tissues, and very likely is the immune phenomena that the Pirbright group describes is related more to the localization in the lymph nodes, whereas the shedding, I believe, is related more to the localization of virus on the surface epithelium. But time will tell, and I expect that we will have a greater understanding of this in a few years to come. So now we take this concept to a field study, and so through collaboration with our counterparts at the Directorate for FMD in Mukteswar, India, we've been doing sample collections at slaughterhouses in southern India, and these are Asian buffalo, and we've done the same sort of approach using immunofluorescent detection of FMDV in the context of cell surface markers, and have found localization within the dorsal soft palate of buffalo as well. Asian buffalo, that is, not, of course, Cape buffalo. And so this is another way that we have integrated field studies with our laboratory endeavors. Duration of the carrier state is another important aspect that we talk about a lot. And so I really like this paper from Tenzin et al. In 2008, a meta-analysis of laboratory studies that looked at the duration of the carrier state. And this has been a very convenient paper that I've gone back to over and over. And so recently, we've tried to replicate that approach, again, with data from the field from India, with support from our collaborators at University of Minnesota, and found data that to be similar to what Tenzin found from laboratory studies. Again, this approach, similar approach to field studies, is the same one I showed earlier from the Bronsworth paper. And again, very nice when we can find ways that the field studies confirm what we see in the laboratory to give us a unified understanding that is not contradictory. So genomic changes within the carrier state. This is something that I get nervous. I'm stepping outside of my boundaries of comfort, particularly certain people in the audience are much more equipped to talk about this than me. But the point is that numerous papers, or more than a handful of papers, have looked at the concept of whether the carrier state is related to changes in the FMDV genome and whether those changes can consistently be found in carrier animals. This is an example from Parthaban et al. In Satya Parada's lab, and looking at positive and negative selection across the entire FMDV genome. I'm not going to get into talking about data, but the point being that this was a laboratory-based study, and we have been attempting to do similar type of work with field-based study at the population level. And again, this delineates the same concepts of advantages and disadvantages. This is a very small group of animals, but much better control of the samples, whereas here we're looking at population of animals, but without the type of control that we have in the laboratory experiments. Ultimately, where we're going is trying to find these magical events when we think we can show that some lineage of outbreak viruses has occurred from carrier viruses. And I am by no means saying that this is a definitive demonstration of that thereof, but it is what we're after, trying to identify with the population level approach if we can identify those types of events that are so hard to find in the laboratory sometimes. So transmission during the carrier state. This is the same Tenzin paper again that quantitated it to be exquisitely low. That's something that we all know and have seen in various forms from experimental studies. This is the Parthaban paper again, which attempted to transmit cattle to cattle from carriers and did not succeed. This is Dr. Murray's paper that was buffalo to cattle, but again, did not succeed in transmitting from carrier buffalo to cattle. Those were all experimental studies. We have conducted a small study in the field in Vietnam in which we identified carriers and paired them with naive sentinel animals, commingled them for months up to a year or more, and sure enough found no transmission. Gets you to the point that you start thinking, well, how much negative data is it going to take before we're ready to accept that these animals are not a threat? But that's above my pay grade and it's a subject for other people to decide. But that said, I'm closing in now on the end, and so keeping it real, again, I want to emphasize the point that I believe these two sub-disciplines of our work, laboratory and field studies, that they should not be seen as adversarial. They should inform one another. And in my efforts in supervising scientists, I supervise epidemiologists that I'm constantly reminding that they have to understand the pathogenesis of this disease in order to describe the epidemiology. And it goes the other way as well. The laboratory scientists, I tell them, they must understand what's happening out in the real world. And so modeling, as the last word I throw in there, we're in an era that modeling is becoming more and more part of our life for various reasons, and it's incredibly powerful techniques, but I would make the same point that I think modeling has to be informed both by field studies and the laboratory experiments, all of which can contribute to a more holistic and comprehensive understanding of the carrier state in both settings. So with that, I'm going to close out and thank my co-authors, Dr. Rodriguez, Dr. Stenfeld, and Dr. Brito, international collaborators that have helped, that we have strong collaborations over the years, Dr. Zung and Dr. Long from Vietnam, Dr. Ranjan and Dr. Jitendra Biswal, who are here today as well. We've had collaborations with University of Minnesota, with Kimberly Vanderwall, who's here, as well as Andres Perez and Shiv Hayer, who've contributed to some of this work, and Amy Delgado at SIA, APHIS. And of course, I want to recognize EuFMD and JIFRA for giving me this opportunity to speak. Funding comes from our USDA ARS base funds, as well as support from the Defense Threat Reduction Agency of the Department of Defense, as well as Department of Homeland Security of the U.S. have contributed to some of this work along the way. And again, recognizing my international collaborators at DAH Vietnam and DFMD of ICAR India. Thanks very much.
Thank you so much for sticking to the time. That was perfect. I'm going to allow one or two questions. This is really exciting work, and I think, yeah, lots of reasons to get excited about. Satya, and then Mariana.
So thank you, Jonathan, for a nice talk. But just this is not a question, but it is a comment. I think in 2006, already we have published about the detection of carrier animals by using the saliva samples. And later on, every 2 years, we have presented in all the EuFMD meeting how we have developed these carrier animals detection by saliva, nasal, and probang sample for different serotypes like OA, and this time we are presenting Asia. And we have developed recently a kit for OA and Asia, which we are presenting tomorrow. This is very nice. I am very happy to see your data that coincide with our data. So this is very good. But regarding the detection of a carrier in 28 days or 21 days or any below that, so I don't agree any specific day for detection of carrier. What we have shown in 2006 paper, if the vaccinated animal, they doesn't produce any IgA. And if the animals are acutely infected, on those days, 3 days, they produce little bit of IgA. They are very higher than the normal level of IgA. And if the animal is going to be a carrier, they continue the IgA, then onwards. And if the animal is not being a carrier, then it is dropped after 3 days. That also we showed in 2006 paper. So if you think by 10 days, when there is no acute infection, so if you think also instead of 21 days, you are taking 10 days or 14 days when there is no incubation period is over, so you can detect also the carrier animals. That's all. Thank you.
Yeah, I agree with everything you said. What else am I going to say, right? The IgA, I mean, one might question whether that's a cause or an effect of the establishment of the carrier state, and I don't quite know the answer to that. I agree with you that under different conditions, those lines that we have drawn at 10 days or 21 days, those are likely to change. It's a circumstance of the conditions we use. My point is just to show that the definition of 28 days that we all hold on to like it's something so concrete, that's not so concrete in itself. And these that I'm presenting now, they're a little bit more concrete because they're based on experimental data, but I agree with you, they are still somewhat arbitrary and subject to fluctuation.
I think Mariana has a question, but I see that Keith also has a question. I think it's Keith, is it not? Keith, do you have a question? Or?
I'm sorry. Thank you. Thank you. Would you like to speculate at least on whether animals at the end of, when they are no longer positive by probang or by PCR, are sensitive, acutely sensitive to reinfection with the strain for which they were originally carriers?
You know, you need an immunologist for that question, but if you put a gun to my head, I'd say they probably are going to remain protected against the homologous virus for some period of time, yes, but using different parameters to predict that we've been talking about all day and others. That's as far as I'll go out on the limb.
I think then I'll give Mariana a chance as well. Sorry, Mariana.
Sorry, coming.
Thank you very much for your talk. I think I remember that in your paper you mentioned that in one of your last papers that actually the percentage of persistently infected animals was not affected by the fact that they were previously vaccinated or not. Is that correct?
It was very similar, yes, under those conditions.
And does that affect the length of carrier state if they were vaccinated or not? Have you tried tested that? If the length of a carrier state was influenced by the fact that the animal was previously vaccinated or not?
Yeah, in the laboratory, we can never do that test because we can never keep animals for 9, 10, 12, 14 months. We have looked at that through some of our field studies in India in particular, and we did stratify by vaccination status, and my impression is that there was not much difference in the duration of the carrier state. But I'll get back to you with a more definitive answer on that. We also looked at stratifying by clinically affected versus asymptomatically infected animals and did not see substantial difference there as well.
Okay, yes, thank you. Then our next talk is by Nick Knowles.